So this might be more for Ross, especially on the CB1 project that you had. So you were looking at, functional antibodies that had, so we would say agonist or antagonist activities, and in this case, antagonist. How do you? Is there some sort of a selection or filter stage where? So you have a lot of these antibodies that might engage the target, but you really want something that has some functional relevance, some functional activity. And I was wondering, instead of just taking everything that engages and then running it on a flipper or the calcium assay, is there some sort of selection or filtering process that you would do in between there, or do you just have to try them all?
I would love an intermediate step there. It's a giant pain in the butt to clone and express 100 antibodies, and you need a fairly large amount in these functional assays. That's one of our bottlenecks, is you need a fair chunk of them. So we can't seem to predict which is the magic one, and usually it's like a couple of percent of the antibodies are the ones you want, and so you just got to grind through it.
So, for both of you, I was wondering if you're seeing any interest in multi-target antibodies, and then so a lot of the talk has been about specificity and selectivity, but there's always a sort of tides that change a bit in drug discovery. Sometimes it's really kind of in vogue to have something that hits a few things, and then everyone wants specificity. And so I was wondering if you've seen any interest in multi-target, and then in addition to that, if you see any interest in simultaneous multi-target binding and targeting.
By multi-target, you mean independently binding multiple molecules? Or
Yeah.
Multi-subunit protein?
Either, and then also multiple target, yeah. So
Yeah.
for the multi-subunit protein, that would even that would somewhat relate to, say, if you have some sort of a dimer type protein,
yeah.
right? And so either one, but more specifically two different proteins.
We have done quite a few multi-subunit things where the more than one protein make up the molecule, and one that's quite difficult is that is the two partner proteins are expressed in other complexes, and they want to target only one of the complexes. So you've got to hit the junction between them to get that specificity. But we haven't had any programs with multi-target, hitting multiple targets at the same time.
So that's actually this is some very interesting research drug development activities. You just feel why is I can give you a couple of examples. Why is that genotype has a very interesting technology called 2-in-1 antibody. What you do is you take an antibody that's say targeting HER2, right? The tyrosine kinase receptor, let's say Herceptin, as specific to HER2. And then you take the HER2 antibody, trastuzumab, then you begin to mutate the heavy chain CDR region of that antibody. You mutate the antibody, you ask the antibody to continue to retain the HER2 body activity, but in the meantime, to gain body to other targets. So they did that successfully. So HER2 EGFR, HER2 HER3, so they call it 2-in-1 antibody. So antibody by definition, they are monospecific, and you can engineer them to target multiple targets. The same antibody. So that's antibodies made all the way to the clinical trials. No drugs yet, but that's one way. So the idea that is you want to target multiple targets at the same time. So a much easier way to do that actually is the construct called bispecific antibodies. So that is already many drugs in clinical use today. That is, you can make a one antibody targeting, say, HER2, another antibody targeting HER3. They are monoclonal antibodies. That through engineering, you can make this antibody to bispecific. The one arm antibody bind to HER2, another arm bind to HER3. So these are became very popular. Our lab is working on this. And even more complex, you can make multi-specific antibodies. And one antibody molecule, you have the variable region bind to two antibodies. is carcinogen, the C-terminus heavy chain. You can add additional antibody targeting the third or fourth targets. So this is this actually became very is practical, clinically practical platforms called bispecific or multi-specific antibodies. Another way to do this is even for the same antigen, you want to target multiple epitopes. We have one example that's success is HER2 again, hertutumab and pertuzumab, two different drugs targeting same antigen, same protein at different epitopes. They achieve different efficacy. So these two antibodies mostly actually is used as a combination, and particularly for infectious disease like antiviral and antibacterial, and also in cancer. So FDA is open to the idea that you can make antibodies targeting different targets. You can combine multiple antibody mixture. This actually you can develop say 3 different cell lines producing monoclonal antibodies. You mix these cell lines into one mixture and ferment in one tank. So that's called polyclonal monoclonal antibody mixture. There's a company called Safety, which is very, so initially, so you're an engineer, you're back there, so we thought this would be technically difficult because how do you control the bioprocess that the different component of antibody give you the same titer, but somehow engineers has magics that they can actually control this. So this actually basically address your this is a very practical. People use this for targeting multiple targets at the same time.
A question for Dr. Ross regarding the so at very early stage for antibody discovery against memory proteins, we know that the corrected conformation, native information is very important start to get functional antibodies. So it seems that in your slides you mentioned a lot of the lipoparticles containing high concentration of native memory proteins, and you prefer to use DNA prime and then protein boost to get a high titer of the response. So for some proteins, especially the multiparse memory proteins, it's really low expression. So how do you produce these lipoparticles with correct information and high concentration containing? And besides this strategy, are there any other strategy to produce these native memory proteins for us to study for antibody discovery?
Every protein I feel there's a different solution. Like it's often difficult to predict what's going to work. Often we'll look in the literature, and many academics have already solved the problem for us, and we just, but it's very diverse. Like we'll do things like, our expression plasmid is special. We have a special expression plasmid that's very potent at expressing things. We'll try different leader sequences. We will chop bits off that sometimes have trafficking signals, negative trafficking signals that hold it back. We have various things we stick on that help our positive trafficking signals to boost them up. And some of our most intractable ones, the ones that we're stuck with, what we resort to is shotgun mutagenesis, which is basically we bomb it, we mutate every single residue, and we screen for expression. And we've had to resort to that for some cases too, to get expression up. That is a key problem that we often struggle with, and we can spend many months just solving the expression problem in order to move forward with.
So are these lipoparticles generated in vitro, or you need cell fermentation?
Oh yeah, sorry, I didn't answer your. Yeah, live particles are expressed in mammalian cells, and we express them there. So we've got to get them to express in something like HEK cells or something like that. So it's got to. We often actually use not just humans. HEKs are human cells, but often we'll use a a chicken cell line, because a lot of proteins they're regulated at trafficking, and there will be negative trafficking factors that will be grabbing it and holding it inside. And when we some cases we'll put it in the chicken cell and it will shoot to the surface just fine, whereas in the human cell it's stuck inside. So really the short answer is we try a whole panel of tricks to solving expression problems.
In recent years, antibodies actually became very successful in targeting enzymes. So traditionally you have small molecules are very good inhibitors of the enzymes, like for drug discovery. So for some enzymes like MMPs, literally intact proteases, it's very difficult to get specificity. And these are very good drug targets. Recently, there's major progress in that use antibody targeting enzymes, including like arginase, MMPs, and so with what Russ talked about, multi-span difficult proteins enzymes. So we can really expand the target of antibodies.
The possibility of developing the antibody for treating the bacterial and fungal infection disease. So as we know that currently for bacterial and fungal infection disease, we mainly rely on the antibiotics and the antifungals. So so what's the major challenge, the barrier for developing the bacterial or the fungal specific antibody? So do you have some suggestions?
Yes. One good example is Genentech actually has a phase 2 program for drug-resistant staph aureus infection. And so there's a very good paper I actually alluded briefly in my introductory slides. And they have a bacterial surface protein antibody, staph aureus protein, and they have an antibody. So this protein, the antibody bound to this protein, this can be internalized, which many proteins do. And once internalized, this antibody conjugated with a very potent antibiotic, and that's bacterial sandal. And what delivered inside is a bacterial cell. So by the way, this particular bacterial sandal small molecule toxin is so toxic you cannot give to a human, even though it's an antibiotic. So what's delivered inside, so when the antibiotic conjugate circulating is blood, is not toxic, is conjugated, it's a prodrug. What's delivered inside the bacterial cell is a bacterial specific protease can cleave the antibiotic, release it, and target the intracellular target of the bacteria. So this is a system called AAC, antibiotic conjugate. So you differentiate from ADC, that's antibody drug conjugate for cancer treatment. And so that's, to me, is a very exciting development for treatment of bacterial infection. In terms of antifungal, I have not to say any therapeutic antibodies for antifungal. Maybe I missed anything. Do you know any people working on that?
I didn't. Yeah, I didn't find it.
I think it would be difficult for antibody to clear a fungal infection, so that's I'm not familiar with. I think it would be just very challenging.
Thank you.
I think one of the real bottlenecks is really a business bottleneck. That is that the industry is obsessed with cancer, and that's where all the therapeutics go. And it's particularly immuno-oncology, the new exciting field, is, in the last 5 years, there's been a huge shift. Most of the therapeutics are going to cancer. I've got some background in developing, things for fungi and bacteria, and I think there's a huge unmet need there. And even people who make antibiotics are struggling. The business case isn't really there. That people aren't don't see the business case, so they're not going to develop them, and I think that's the real problem. I think the real answer is vaccines. Vaccines is cheap, and that's the way we've got to go, but yeah, I think antibodies, it's a struggle to get people to fund it.
My question is about the use of artificial intelligence and deep learning in predicting off-target bindings or epitope selection. To what extent the drug discovery industry is exposed to artificial intelligence now, and what about the future potential it holds?
Could you repeat the first part of your question?
About the use of artificial intelligence and deep learning, the sequence homology data, a lot of validated data on the internet. Like, how is the industry now incorporating this binding data to predict off-target bindings, epitope selection and binding with this antibody and antigens?
But let me tell you a little bit about my experience. That is, one thing we can really predict is that you give antibody to protein, it's basically a protein, it's immunogenic, as a drug to a human. So we would normally generate immunogenicity, immunoresponse, right? So called anti-drug response. So even through humanization, even the CDR region that you cannot mutate, you can generate immune responses called anti-idiotype response, right? So this, so what you can there's a standard program procedures that we can follow. That is, we can predict B cell, also T cell binding site epitopes. If there's no, the sequence is cannot find in human, then most likely will be immunogenic, so you can mutate it back. So that's, but in my experience is mostly you can do all these predictions, but at the end of the day, you still have to test in animal models, in clinical safety, preclinical safety, even phase 1 clinical trial, this safety study.
I can make one comment on artificial intelligence or machine learning. I mean, one of the holy grails in the antibody discovery world is, can you predict from the sequence what it binds to? And the problem with that, the challenging problem is that really the business end of an antibody, the heavy chain CDR3, is very diverse in length and sequence, and it's been very challenging to predict its structure. So I know there's a lot of people trying these things, but what we would like to do is that, there's this, next generation sequencing. It's very powerful. You can immunize an animal and then basically get all the sequences out, and then you've got 1 million sequences in front of you, and it'd be wonderful if you could pick out the ones that bind the antigen. And there's a few papers out there that do things like frequency analysis, like the ones that expand under immunization, like George Giorgio has published on this, and others. And and he's tested those, and they do indeed bind. And the weakness of that is that it's based simply on frequency, and it only gets you a handful of antibodies. But what would really be nice is some kind of machine learning that could predict the structure of that based on sequence and predict what it binds to. And that seems to me still some ways off, but I think, we'll see. That would be very powerful indeed.
These antibodies can go to the brain? Yeah.
You mean brain penetration?
Yeah, brain penetration.
How they engineer antibodies can use the brain?
Yeah.
Yes, okay. So, there's actually there's one antibody recently approved by FDA as a biologic antibody targeting the beta peptide for Alzheimer's disease. So that antibody is effective. So people still do know why it worked. So the two possibilities. Why is the Alzheimer's patients, the brain is compromised. So the BBB is not intact, so antibody can get it in. Another thing is this actually is an experimental validation. That is the beta peptide actually can leak from the brain to in circulation. So when you have antibody, your circulation can actually bind to the beta peptide depleted, create a sick. So more beta peptide can diffuse into circulation. So that's another way to interpret. This is the more plausible way. So in order to get antibody really inside the brain, you need to have a really targeted engineering. One example I can give to you is actually we're testing in the clinic is this. So the brain has need a lot of carry a lot of redox reactions. So that's mediated by ferric iron molecules. So basically, so this transferrin receptor is one of the most common receptor of BBB. It is very effective to take the iron, ferric iron from the blood, flip in between ferrous to catalyze the redox reaction. So take advantage of that. So origin type data is to engineer the antibody that bind to the transferrin receptor, but not interfere with iron binding. So it's a different part of the receptor. So you create so when the antibody bind to the receptor, that flip in very efficiently. The receptor does not recognize this iron has smaller protein. The ones inside the brain, this antibody is bispecific. The other arm antibody targeting of target of interest. In that case, the example we showed was gamma secretase, which is one of the protein enzymes that are related to Alzheimer's disease. So there's other proteins in the brain. Membrane can do the same thing. So actually, my group has an active program. As you put the poster in my group, actually engineer antibodies trying to penetrate inside the brain. We have an active program on Alzheimer's.